a larger class this time. That okay? All right. So maybe in a week of time, and just during the past week, next week, send an email to Sean about your group assignment. Okay? But I want to each group has at least 3 and most 4 people. Right? If you cannot find more than 2 people are willing to work with you, and then let me know. Okay, and then I will assign you to some groups. All right.
Great.
Any question about? Oh yeah, another thing about the exam. All right. So we went through. I'm talking about the exam. We went through the answers last time, and I hope you all agree that the answers are right. It could be wrong. That's fine. And then, but we will not return your paper back. Okay. For the reason that I may want to reuse some of them as well. So if you say, oh, I should do better than this. I want to check my score or whatever and things like that. You can do that. But here's the catch. Okay. It's like play football, and you throw a red flag. All right. If you win, you say, okay, Sean made a mistake when they graded. If you win, you get the credit back. Okay. If you lose, it will deduct 2 more points. So you are adding a risk. Okay? Otherwise, everybody goes to him and say, okay, I want to get any single point. But again, don't worry about the overall score. There will be plenty of opportunity to rescue your miserable failing midterm papers. And actually, I don't know, sure probably haven't talked about that yet. So in the RNA sequencing part, we may have some bonus point available to do another level of homework or things like that. So there will be plenty of opportunities. Don't worry about that. Okay.
Mm-hmm.
Recording.
Yeah, sure.
Did you? No, you're in trouble.
So, all right, let's start. So let's talk about something fun. So we don't need. So we have we have talked a lot of algorithm stuff, and we have talked about of method computational modeling, and we get through some very detailed statistical procedure in the past hands-on part. So now just talk about something new, and something just a topic named single-cell RNA sequencing, without with a lot of debating, with a lot of discussion and promoting area, and which I'm working on. So, and this part, because it's relatively new, so a lot of things are uncertain there. There's no very detailed claim which method is better. No such analysis. Very little analysis about that. And also a lot of methods are just appearing and under developing. So not a lot of fixed answer, a lot of open questions here. So please just open your mind and just think if you are interested in this part, think about it. And so because no fixed answer, so no final exam questions, or no, at least not a lot. Yeah, yeah. Yeah. Yeah, not a lot. Yeah, let's start. So background of single-cell RNA-seq is quite straightforward. It's just single RNA sequencing profile of the transcriptome from single cells. So we want to get is just get one single cell. Can we get the transcriptome measure of this single cell? So the first work of single-cell RNA-seq sequencing, people may regard as the first work was from 2009 as about this work. So RNA-seq whole transcriptome analysis of single cell. It just shows the capability. Okay, we can do RNA-seq sequencing, RNA-seq for single cell. But most the high quality single cell high quality single cell RNA sequencing data appeared. The first two works appeared in 2014, all by the Dr. Aviv Ragev's group from Broad Institute. So almost the same time, one on Science, one on Nature. So one is about the using single cell RNA-seq data to study the immune response treated with certain stimuli. And the second is about to study the intratumor heterogeneity, or the heterogeneity of tumor tissues of glioblastoma. So the characteristics of single cell data is that because it's low input, the number of the sample is low, and also the amount of sequencing, the amount of gene expression you can sequencing result you can get is not that large comparing to the bulk data or bulk tissue data, bulk cell data. But you can do it for a lot of cells. So we just change the way to get really deep sequencing of a small number of bulk samples to get to relatively low sequencing amount for a lot of cells. And this is my idea of single cell data. So that's why I regard these two papers as just as a milestone of single cell RNA-seq data. RNA sequencing is that in these two works are the first works that really gave more than hundreds of cells measured from one experiment. And with the sample size is enough to really do some modeling and the biological mechanism level study. So let's start from why do we need a single cell RNA-seq data? So because sometimes one most straightforward thing is that we just compare the difference between single cell and bulk tissue. So in some bulk tissue, for example, like cancer tissue, as shown in this figure. So cancer tissue is composed by cancer cells, immune cells, and a lot of other cells. And when we want to study the real mechanisms, we may need first to observe the bulk tissue sample. It's just the average sum of the signals from different cells. So, but the different cells, each of the characteristics of the cells really highly determine the disease characteristics like progression and also the metastasis, and also some drug resistance. And but this is just part of the mechanism, but not all of the mechanism. And so the first application of single cell RNA-seq sequencing data is to study intratumor heterogeneity or intra-tissue heterogeneity. For cancer study, to study immune response of a blood sample. Blood sample is still blood tissue, and still with heterogeneity there. And also developmental process, like for the development of like brain tissue, and with the cells at different layers have very different behavior with some spatial information there. We will talk about it later. And also one underlying assumption we have, or underlying principle we're using here to think about the bulk tissue sample. Bulk tissue data is about the hierarchical heterogeneity or hierarchical transcriptomic heterogeneity of the tissue data. Actually, this is my definition. It's not the common definition. So I just want to introduce this idea is that first, different cell types may have different gene expressions, but the different cell types may have quite distinct expression profile. And this can be controlled by the epigenetic level regulation that constantly regulates the difference of the cells. And also, even for the same cell type, for the same immune cell type, for the same cancer cell type, cancer cells, may not be same cancer cells, but for all the cancer cells, of the same or similar cell types, the gene expression may also show difference. This is altered. Actually, the gene expression is controlled by the current regulatory signal in that cell, in each cell. And for the cells at different environment in the tissue, the cell may have different regulation input. And the different regulation input may result of different expression profile. So in sum, it's like the different transcription regulation effect can be epigenetic level regulation, can be transcription factor cis regulation, can be mutation of certain promoters, also mutation of genes, can be microRNA, can be a lot of that. But in reality, in the real case, we cannot measure these factors simultaneously from of a same cell. But what we can do is we can observe by using single cell RNA sequencing, we can observe the gene expression profile of each cells. And each of the expression profile is a result of the current regulatory input. So with this truth, we can definitely do some computational assumption. We will see it in later. But the hierarchical heterogeneity is that there are some, for the same cell type, they may have some the regulations, some of the transcriptional regulatory signals may regulate the expression profile, the gene expressions in each of the same cell type, and this and also for the different cell types, there are also like a higher level or we can term it a higher level or lower level. I think it's the different level of regulation, heterogeneous regulation mechanisms, like the transcription factors can regulate the more flexible or like more or less constant expression changes, and the epigenetic regulators can regulate the more constant expression changes. So and also this is because I say it's new is that I will talk about there are some single-cell RNA-seq data-based transcriptional regulation study. But in the past, we started transcriptional regulation event just from the idea of, okay, who regulates what, like chip-seq data study, and just to study the regulatory element and the downstream effect. But in single cell, single cell really give us a capability to think about this problem from this question from another angle. Is that the specific characteristic of single cell is that each single cell, the current expression of this cell is purely regulated by the gene expression signal, by the regulatory signal of that cell. You really don't know it. But for bulk tissue, we don't have that because the different cells in a bulk tissue may have quite diverse or heterogeneous transcriptional regulatory signal. So this is one possible. This is just one possible application. We may talk about more details later. And the second one is about more straightforward. It's easier to think about than this one, drug resistance study. So we may just think about cancer. So for some targeted drug. So for example, it's just like for leukemia. So there is one mutation, very common mutation named FLT3. So the FLT mutation may cause it's a tyrosine kinase may cause consecutive stem cell self-renew and cause the leukemia formation. And there is a drug there's a drug to inhibit this mutation. So if everything is okay, so this drug can just definitely treat the disease. But for some patients, they may easily develop resistance. And the one thing to develop the resistance is one mechanism is that we have one mechanism, especially one observation we have is with the concurrent mutation, if the patient has another mutation in TET2 or DNMT3 gene, this patient may easily to get a resistance. And then the researchers checked about what is the function of TET2 and DNMT3. This is a methylation regulator. It's not just the function is totally different to FLT3. But then people found so okay for the post, and because we can there are real patients we cannot do any manipulation, but we have in-lab mouse model. So we do it on mouse model, and from mouse model we found okay for the mouse with the concurrent mutation, after we treat the disease with the mouse with the concurrent mutation with the FLT3 inhibitor, and for the mouse develop resistance. We found, okay, there are certain transcription factors activated in the residue of the resistance residue, but the transcription factors are not consistent through different replicates. So then people may write this hypothesis. Because the TET2 and DNMT3 are epigenetic level regulators. So with lots of them, so the cells may have more variability or variability of the epigenetic regulation, and increase the potential of the cell just by random develop activation of certain transcription factors can evade the treatment or can get the resistance. And how to test this? Then get this background. Get back how to test this. How to test the epigenetic. If this is correct, if there's a certain way, we have found that okay, certain transcription factors are really popped out or really developed in the residual samples if we just test the bulk tissue sample. But how to confirm? Okay, it is the epigenetic level heterogeneity really cause that. So most straightforward way, okay, we can do it. Is that what we get the single cells we get the single cells. We don't do RNA-seq data. We do single cell epigenetic data to see the methylation level. Single cell bisulfite sequencing data to see it. To say if the pre-treatment is with more heterogeneity and post-treatment with less heterogeneity. But the single cell bisulfite sequencing data is currently is more expensive and it's not that ready compared to RNA-seq data. But we know the RNA-seq just can reflect the transcriptional signals. So we just test. The second way what we can do is we still do the single cell of pre-treatment and post-treatment residue, the resistant residue. And we generate RNA-seq experiment form of it. And we see, we use computational modeling to see if the regulation or the co-regulation modules or co-expression stuff of the pre-treatment and the post-treatment shows distinct difference. And the difference is like the pre-treatment, some samples may form a small co-regulation module, some others may form small co-regulation modules. There are a lot of small co-regulation modules in the pre-treatment cells. That means the cells are really rich, more transcriptional regulation heterogeneity. But for the post-treatment, the co-regulation modules are larger. That means they are more homogeneous. So by using this method, this kind of ways, this is a real case. It's not that theoretical, but at least by using this ways, we can try to see, okay, we can try to confirm, okay, there looks like more possibilities that at least the transcriptional level, regulation level heterogeneity contribute to the resistance development. Because this is a real case, not that theoretical correct, but at least this is a solution. But what I want you keep in mind is that for some hypotheses, or for your biological hypotheses that are really related to the cell level, intra-tissue heterogeneity, you can check it by the single cell technique. And the single cell RNA-seq technique is currently the most mature single cell omics data. So and can reflect some activity level changes and also some regulatory level changes. And also the last example is also a real example. So actually one of my collaborators, so she did the single cell RNA-seq data on cell line. So the first time I met with her to discuss about this, so why you did it on cell line? Because cell line, we know the cell line are most of the cell line with the same treatment, they are quite homogeneous. But why do you do this on cell line? Then she told me, so the reason is that we do shRNA to knock down one gene. But for the shRNA, if it's for some of the cells, it cannot be fully knocked out. And if we treat it too much, all the cells will die. So they want to get a balance point. For some of some of the cells are with this shRNA successfully knocked down the target gene, and the cell is still alive. So they want this point, so they do single cell RNA-seq data to get if they can get for the shRNA group compared to the wild type group. The first they want to see if there are more cells, significant more cells with that gene is knocked down by shRNA, and also extract or sort out the cells from the experiment with this gene knocked down in the knockdown group. So these are some possibilities the single cell RNA-seq data really grant us. So there are some really high-level high-level publications about single cell RNA-seq data, especially in the beginning of the emerging of the large-scale single cell RNA-seq data. So, like I mentioned, one possible application of the single cell RNA-seq data is to study the intratumoral heterogeneity. So this work is published in 2015 or 2016, I think, but still by Dr. Aviv Rakev's group and also Dr. Levi Garvey, who is in Lilly right now. So at that time, he's at Harvard, Dana-Farber. So, in this study, so what they did is to get this number of melanoma patient samples. We can say it's like 13 samples. And for each of the samples, they measure 600 to 800 cells. And this time, they didn't do cell sorting. Just to get most of the cells in that tissue. So they can get malignant cells and also non-malignant cells. So they use certain markers to sort out after they get the gene expression file, they can say, okay, which are the malignant cells, which are the non-malignant cells. They find the cells from different patients of the malignant cells are truly clustered into certain groups. That means this is not intra-tumor heterogeneity. This is inter-tumor heterogeneity. That means among different tumors, they have big difference. And this is for the non-malignant cells, immune cells, NK, natural killer cells, T cells, macrophages. They found, okay, the cells from different patients, they are really like massed together. And although there are for some specific cells, they may form some subclustering, but they may still like just capture the immune cells. The immune cells are closer to each other compared to the malignant cells with respect to the inter-tumor heterogeneity. But with this, at least they can just characterize the intra-tumor heterogeneity by using this way. And this is a TSNE plot, like the PCA MDS plot I have introduced to you. It's just use the 2 dimension to reflect the data, how the data looks like. And we regard one word I have always talked about is the data parasite. So for us, data parasite, we analyze this data and get some more results from the original work. I may show you one result later. And the second work is about this is published in 2014. It's among the first parts, the first generation of large-scale single-cell RNA-seq data. So still by Dr. Alvira Raghib School. So this one is what they did is they get the mouse dendritic cell and they treat it with LPS at different conditions with different time. They get the time course data. And they found that, but please keep in mind, so this time and this time the cells are not the same cell. So for single cell RNA-seq data, once you use the cell, it's used. So there's no way to test the cell 2 times by the current technique. And what they found is that, so very interesting thing is, I think at that time, currently if you get this result, people may challenge you, but at that time when the single cell just appeared, this is really a very, very significant result. They found for some genes, the x-axis shows the time point, each dose is one cell. Y-axis is the level of certain immune response. They found that at the 1 hour treatment, there are certain cells that are first activated first. And then their hypothesis, the hypothesis raised from this paper is that with certain mechanism, certain cells stimulated first, these cells may have impact to the other cells to stimulate the whole immune system. And this is just definitely a single cell. By that data, they cannot study how if this one can really affect the others because they don't have the one cell, they only have one data point. They don't have they cannot see one cell continuously or with multiple time points. So they cannot fully answer that, but they see some pattern. And also there are some, so just from my personal point of view, the best application of single cell RNA-seq data is not in tumor study, it's not in cancer study, it's in brain science study, it's in neuro study. So there are a lot of studies about about the mouse cortical cells, about the mouse cortex cells, and about the brain tissue and brain development. And Dr. Linnerson is a really big guy in that field. His group has generated a lot of high quality data. But this example is not from his data. So it's that what they did is they did very fancy cell collection from the different spatial coordinates of the brain. And because the brain, you can do it for the different development stage, you can do it for the different spatial distribution, and you can do it if you can get the cell. I will talk about the single cell RNA-seq experimental protocols later, but if you can get the cell, you can get the information, you can test it. And then this technique enables you a lot of capabilities to really test how the cells at different spatial coordinates or at different time varied and how they relate to each other. So this figure shows this figure shows at the different layer of the neuron, and the cells may have very different characteristics, and they get just for a specific biological process or related to the neural response, something specific to neuron, and to get a path of that, and they can make certain biology from that. This is very cool and very fancy study. So and one project I would like to introduce about is the Human Cell Atlas. This is an ongoing project. It's still calling for like computational grants, and also they have granted the first the first cycle of the the core groups, 38 groups among the over the world to study the human cell atlas, to study the to conduct this contribute to this consortium project. So basically, the mission is to create comprehensive reference maps of all human cells and the fundamental units of life as basis for both understanding human health and diagnosing, monitoring, and treating disease. One thing very important to be answered here is because we want to build a reference map of cells, and also we want to also we want to link it with existing tissue data and how to use it to get our current understanding to current results from the past tissue-based database study. And also it is worth I really want to note here that our IU campus, the current the Center for Medical Genomics, we are already generating our single cell RNA-seq data here. So and for some specific cell types, cell lines and primary cells, and and there will be more possibilities in the future, definitely. And the cells generated here are majorly just guided by the different research groups, by the PIs who generate the sample. So the cells may be very different to the major cell types in the human cell atlas. So we will have some of our own streams of our own data of specific cell types, just maybe in complement to this cell atlas project. And now I may talk about the experimental protocols to get single cell RNA-seq data. So getting single cell RNA-seq data, I think the more RNA-seq is still RNA sequencing, as what I'm talking about. But the trickiest part is how to get a single cell RNA sequencing, how to get the samples from single cell. So one kind of protocol is named the underlying idea is get cell sorting. We sort out certain cells. We get some certain cells, and we sort out the individual cells, and we construct the sequencing library for each of the cells, and then do the sequencing. And one example is C1 Fluidigm Smarter pipeline. So Smarter is the sequencing pipeline. It's just a library construction pipeline. It's a very sensitive way to extract the small amounts to construct library for small amount of input RNA. So here let me show a video about how this fancy stuff works. So let me put it. Next time I should get a mouse. This happens a lot of time. Yeah. So here we go. So just start over. Yeah. So this one is just the array of C1. So it's very, very complicated thing, but the flow is like that. The cell flows into the array, and each cell will be stuck here or will be captured here, and the cell will keep flowing, and this can simultaneously get 96 cells for experiment, and for each of the captured cell, the next step will the captured trap, they will get the cell and get one cell there, and then construct the library from the cell. So the downstream including oh sorry, I just stopped here. So and the cell will go into the next step, and will be then the next step lysis just breaks the cell into the RNA samples, and then they may use the SMART technique to do the library construction, to get the RNA-seq and get the cDNA, and get reverse transcription, and then get the amplification. So this video is, I provide the link there so you can watch this, because this is very cool stuff. So currently the CMG here is using the this C1 protocol. So and please keep in mind here, each of the cell will get into one library for sequencing. And the second and the advantage here I want to talk a little bit is that keep in mind, I will talk more about this later. The advantage here is that for the C1 based studies, you know which cell, the cell phenotype information. You can control the phenotyping information. You can do cell sorting by using certain markers. You can get what cells you want. And the second one is, but the cost is very high. You may need to construct one library for each cell. And the cost of library is currently the most expensive part in single cell for this protocol. And if you, the current price is $21 per library in CMG. So if you really want to get 1,000 cells, it would mean like $20,000. You may need to spend that. But the second kind of just before sequencing, not even includes the sequencing part. Yeah. And the second part is about the experimental protocols. The second experimental protocols is about the droplet-based methods. So basically, this I want to use one 10x genomics chromium system as an example. So basically, this kind of method, underlying idea of this method is that we get the cells into, by using certain ways, we get barcoding the transcripts from the same cell. And then with the barcoding of the transcript from one cell, we can mix the cell, all the samples together and all the sequences together, and before we sequence it. And then we can use this as a whole library to do sequencing. And after we sequence it, because we know the barcode of the transcript, the race from the same cell, so then we can say, okay, this race are from this cell, this race are from the second cell, this race from the third cell. And then with one big library, we can measure a lot of cells. But, and here is the text genomics chromium system working flow. So I got this from the original bioarchive paper. So and this, let's see this video first. So about the text genomics flow. So not very, this one is not. Oh yeah, and also because we don't need, we need this protocol, we need this protocol, I think the most important thing here is about the solution, right? So each time the agent we use here is much more expensive compared to the C1 protocol. So they demand the chromium solution. And start with hundreds to thousands of cells, and with this system, and use our sequencer here. We can be linked with our sequencer, and also because we are bioinformaticians, we don't need their software definitely. So and then we can just go from the pipeline. So and what they use is called the gem system, and in their solution they can form the gem beads like in this way. So this is one cell, and this is one gem bead, and this is artery, and you can, and then you can, with each of the beads there is one cell, and you can link just one specific barcode and the UMI and the poly-T, this kind of sequence or adapter to the bead, and then with the pipeline is kind of, this is the instrument, this is the original cell, where the original cell is, this is the final cell will get out from the oil well. Yeah. So basically they may, this is each bead, each gem, and for some cells they will be linked with one. This is called just get linked with just a small not adapter, but just the barcode, UMI, and the poly-T adapter there. So and then through the reaction, they can get for all the transcripts from the same cell, they may get the same barcode. This is the key idea. And for the downstream amplification, and this is the final race which it may looks like. This is read one. This is read one to parallel read, and also with the with barcode here for the cell to enter the cell, and UMI just what I have introduced in the first class is just to mark this transcript. And then using sequencing, you can sequence it, and then you can also then you can call the genes from each cell. But please keep note here. With this procedure, we don't know what is the cell, what is cell one, who is cell one. We don't know which cell type is cell one. We need to infer from the data. And just here, yeah. And this is the analysis flow, and they extract the barcode, and then they align using star, and also the correct UMI is just adjust by UMI to remove the like redundant transcripts, and also then they sort use the count UMI by they may have a procedure. I think select cell association barcodes and for the gene barcode matrix. So it's like finally you get the mapped for each of the mapped arrays. You may get in this current pipeline. Probably the pipeline is still developing, but for this pipeline you may get each gene mapped to which position and with which barcode to mark which cell this gene is. And then you can build the count table of by the alignment. And
so for this one, you only measure you cannot measure the entire RNA molecule.
Oh yeah. Only three prime, right?
So you want to tell people this point?
Yeah. This is not. Yeah. So this is the next figure.
Okay.
Yeah. So this is not the full length transcription data. This is just the three. This droplet method is just a tree and counting data. Not the.
But if you go back to the previous slide, so if panel D, the small panel D, so that shows that the reason for that is you have a big chunk of poly A part, and then that's your read number one, and your read number two is only going into the RNA a little bit. So you cannot, if you say this is RNA molecule, it's 500 nucleotide, and you cannot possibly sequence the buffer end of it.
Exactly. So this is definitely not for the alternate splicing, something like that. Yes, it's not good. Yeah, but just can measure the most frontier part, so the most forward part. Yeah, and this figure.
That might be an example.
Yeah, example. Yeah. So this figure is very important. It's important. So it measures. This compares the different pipelines of different protocols, and the transfer data, the full length and three prime onion counting this is very important first. This can enable you what to do, what you cannot do. But about like alternate splicing, you cannot do by this technique. So, but also some other things very important is first is about the throughput, the number of cells you can measure by the C1 technique. It's like hundreds to thousands. So because if you do more than thousands, then you need more than 10 of the array, and more than 1,000 libraries, the cost will be extremely high. But I know I heard that I heard a rumor from the Bar Institute. So Dr. Aviv Ragev's group, they are using this kind of pipeline, but working on more cells. But their budget is $2 million for the project. Yeah. So for this kind of the droplet-based method, they can just definitely do like they can do thousands or 10,000 cells simultaneously. And but one thing it's worth to say here is the typical read depth. The read depth of the C1 techniques is like 10 to the power of 6. But for the droplet-based methods, it's just 10,000 to just 10,000 to 100,000. So what's the issue here is that on average, so from a lot of existing single cell data analysis, we may say on average, the average significantly express the genes of each data set of each single cell RNA-seq data is like around 8,000. This is the average number we have seen. So if there are 8,000 significantly expressed genes, and in total we just get around 10,000 genes, 10,000 counts. So definitely the 8,000 genes, significantly expressed genes, are identified by the data generated by C1 protocol. But if we use the droplet protocol, on average, in total we just get 10,000 genes, but we have 8,000 expressed genes. That means on average, one gene can just get one count. So this is the small number of counts. It is okay. So then what it will result for? It will result in that some of the genes with significant expression, you cannot observe the expression. But for some genes with high expression, you can only observe very small amount of the expression from, very small amount of counts from those genes. So, and this is cause one problem is if you want to use single cell RNA-seq data to study some mechanism like co-regulation pattern, like certain regulation variations among the same cell type, the small amount of sequencing read depths here, you cannot do it with this data. So if you want to do, but if you just want to do self-sorting, you want cell type prediction, you want to predict, okay, which cell this the cell it is, like T cell or B cell, this number of rows is okay. So, but if you want to do more mechanism level study, you may definitely need more depth, more second, more counts. This is one key point. And also, keep in mind this kind of technique to get relatively lower average read depth, but more number of cells. So
can you go back? So,
yeah.
So I want to make a point here is for C1 fluidine, and which is the first smarter assay. And then if you look at the read depth per cell is 1 minute, 10 power 6.
Yeah, 10
right?
power 6.
And if we compare with chromium, which is the third column from the last, and that is about 10,000 to 100,000 cells raised.
Yeah.
That is not comparable to each other, right? The number is not comparable because it's not a fair comparison. Because for C1, you are measuring the entire transcripts.
Exactly.
And for 10x, you are only measuring a very restricted part.
Exactly.
So maybe that for the same molecule, you can measure 5 rays out of C1, but 1 out of the extent 10x is
exactly.
still the same thing. So you don't need that many rays in 10x protocol.
Exactly. This is a good point. Yeah, but my point here is that if the average counts per gene is too low, we cannot say, okay, if this expression level can really reliably reflect the current expression abundance, they can just reflect it's expressed or not, but not the abundance. So it's hard to study like co-regulation, some fancy.
I think with 100,000 on the 10x, you can. It's quantifiable. It's not only tell you on or off.
Yeah, exactly. Yeah.
But with lower than that, maybe.
Yeah, it will be a problem. Yeah, exactly. Yeah. And also, then I want to talk about some computational challenges. So here I want to illustrate 3 major challenges. First, for each of the gene, you may think because it's from single cell. The single cell, the gene is regulated by the current regulatory signal. There is empirically for each gene's expression profile, you may get a lot of cells for this gene are with 0 expression. So in single cell RNA-seq data, there are a lot of zeros. How to deal with the zeros? Because you want to do the downstream analysis, the 0 is quite annoying. The error term of the 0. What is the error term of 0? Because for the error term of some values, you can use like Gaussian distribution to model it. We can use like negative binomial from the D-seq to model it. But for pure 0, how to model it? And the second challenge is about the covariance structure of the genes. So just forget about covariance. Just think about look this case. So this is one from one review paper. So that's the cells, just regard the cell cycle. They may at different cell cycle stages. And also there are some early normal cells, there are early apoptosis cells, there are late apoptosis cells. So for cell cycle, just think about it. Cell cycle is one process. Apoptosis process. Some cell functions, something related to cell function, some like in response to different microenvironment, there are also different biological process. Yeah, but the problem here is that for the single cell, the different cells for the different biological process, they may have quite different state, and those different biological process may not be that highly correlated. So that means if you use just the cell cycle genes to do the clustering of the cell, you may get a cluster. So with respect to just the cell cycle characteristics. But just by using apoptosis genes or cell death genes, you will get another cluster. So this problem is that, what this figure shows that is the TCGA data. We get the top and the single cell RNA-seq data. We use exactly the same amount of samples, but when we get the top 5 pieces, top 10 pieces, top 20 pieces, how much of the variance they can explain. So we can see the tissue data can explain much higher, but the single cell data can explain much lower. The reason is that each PC is just in the box cell, a lot of process just are normalized by a lot of the cells in that bug tissue. For example, it's like for the cell cycle process. On average, just two tumor tissues. On average, the average cell cycle signal may be similar, but for the single cells, they are distinct. And the third one is about visualization. Is that the same idea from here? Is that because this big variance among the big variance of different or heterogeneity of different biological process and of different cells. So if we use all the data to embedding or just to map the cells to low dimension and to reflect the cell pattern, if you use different genes, they may be different. So this is third challenge. Just keep in mind these three challenges. And let me quickly talk just 3 minutes. So just quickly guide through all what we did, something we did here. So this is the log RPKM data. So, and these values are all the original zero, and should be gets into infinity, but I just plotted here. So once we check a lot of data, a lot of genes, a lot of data set, we found that here we clearly get a cut, and we still want to fit the data by mixture Gaussian distribution. We can see, we found that here is a gap in this tail. And this part, if we fit this by mixture Gaussian distribution, the frequency here, missing here, should be equal to the frequency here. So we can use this model, just like it's called left truncated mixture Gaussian model, just regard this part as missing data. The data on the left of a certain cutoff, and then we can just write some algorithm to get to infer the mixture Gaussian distributions. And the most important thing is that with this one, so I didn't show the slides here, but we have some good self-fitting statistics can show this method really works well. But the most important thing is that from multiple existing single cell RNA-seq data, we found most of the genes are with 2 peaks. So the figure here, the green bar shows the proportion of the genes with 2 peaks. Light blue bar shows with 1 peak, and the pink bar shows the pink part shows with more than 2 peaks. So you can see most of the genes are with 2 peaks. One is normally one is non-expression, no expression, 0 expression, and the other one is significant expression. And for this part are with multiple peaks. So at least this can tell us, okay, the genes on single cell level, it's more tuned like activate or inactivate. And for someone is with multiple activation state. So then, because we know here is something extra, because with this, first with this formulation, with this model, we can accurately model the log RPKM data of single cell data. We can identify outliers because we have probabilistic model. And the second important thing is that because we found, okay, most of the genes from 2 peaks means activation and inactivation. We just discretize them to 0 and 1. And then we want to partially solve, okay, which biological process or which subset of genes may share a similar expression pattern, may possibly be co-regulated, or may possibly be involved in similar process. So what we do is, this part is quite a little bit extra, but what we do is we use a technique named by-clustering. So simultaneously identify a subset of cells and a subset of genes with a significant high expression or low expression, like shown in this figure. So and may possible, and this is just something extra. So because time issue, we just skip this part. And also I may talk about the two more slides. One slide is this one is now. There's one technique named monocle. So monocle, the original design is quite simple. It's that we still use the PC plot, some idea like that. We use a technique named the TSNE, which exactly the technique generates this kind of figures to map the single cells to 2D dimension based on the original data. And then they try to build a minimal spanning tree. Actually, it's not that hard, but the idea is just they build a tree over the cell. Just build a link over all the cells. To group, first can group the cells. And the second, they can try to build a path of the cells, how cells are related to each other. So this is from real data. So what we have. So here are the 4 conditions: hypoxia conditions, hypoxia knockout conditions, hypoxia and the normoxia knockout conditions, normoxia conditions. We can see this. Just with this technique, we can just visualize the single cells. But for single cells, we have like 4 conditions, and after that, we can say, okay, this lineage, this part is more like the effect of the knockout, the hypoxia, and this lineage is more like the effect of knockout. So, but please keep in mind. So the final key point I want to convey here is that so we can always generate this plot by using all the data. But like I mentioned, for different process, the data variance may be different. That means for each of, you can just randomly choose a gene subset or just choose a subset of genes of certain biological function or biological process and do this analysis, and you may get a different figure. And the different figures, sometimes the different figures may really represent some biological meaningful things, but sometimes it's just junk, it's just noise. So if you use all the genes to do the analysis, you kind of forced to squeeze all the different structure to one structure. But the key point here is how to refine your analysis to get to really start from the original, the big from the original data to get into the reliable or meaningful clusters or trajectories correspond to each biological process that are meaningful there. So this is the current biological challenge. So this is the current computational challenge there. But my lab is trying to solve this problem right now. So, but we haven't get into the final stage yet. Yeah. And the last thing is about there are some published single cell and correlation-based study. So already this was just recently published on Nature Methods, just last month. And what they do is just identify co-expression modules, and then they match it to regulons just to see to find the motifs that may co-regulate these genes and build a correlation network. But we can definitely discuss later about this if you are interested in. But the problem is that the single cell we are using for RNA-Seq may not have the exactly same the motif. So this may be problematic. Yeah. So that these are all the materials I have for this course, for the RNA-Seq part.
Any questions? Go full.